Hello everyone, I am Dr. P. Saujanya. I welcome you all to the lecture on culture media. So culture media is a nutrient material prepared for microbial growth in the laboratory. And this media contains nutrients required and the essential growth promoting factors which enhances the growth of the bacteria. And the requirements in the preparation of culture media are the media must be sterile and it should contain appropriate nutrients and must be incubated at appropriate temperature. These are the requirements for the growth of the bacteria when it is inoculated in the medium. So what is a culture? Culture means the microbes that grow and multiply in or on the culture media is culture. In the medium means when it is inoculated in liquid medium, that is broth. And on the medium means when it is inoculated on the surface of the medium, that is solid agar medium. So either in the medium or on the surface of the medium, whatever the microorganisms that has been grown is known as culture. So the media generally, depending upon the consistency, they are of solid media and liquid media. The solid media, it contains nutrient material containing a solidifying agent. So this solidifying agent, this media, it can be prepared or poured in a plate like Petri plate, or it can be prepared as a slant, or it can be prepared as a deep, that is a stab culture like that. And the most common solidifier which is used in solid media is agar. And this agar was first used by Robert Koch as a solidifying agent in the medium. And the uses of agar is it melts above 95 degrees centigrade temperature. That means it gets liquefies above the 95 degrees centigrade temperature. Once melted and does not solidify until it reaches the 40 degrees. So till it reaches the 40 degrees, it remains as a liquid. And it cannot be degraded by most bacteria. So this agar medium, agar whatever using as a solidifying agent, this will not be degraded by the most of the bacteria. And this agar is a polysaccharide which is prepared from red algae, Gelidium amansi, red algae, Gelidium amansi. And this was first used as a food thickener in by Fana Hesse. She was a housewife and she was using the agar as a gelling agent for the preparation of jellies in her kitchen. And through her husband, Walter Hesse, this agar was known to the Robert Koch. Walter Hesse was the assistant to Robert Koch. So this Walter Hesse came to know through his wife Fana Hesse that agar can be used as a gelling agent. From then onwards till date, this agar is used as a gelling agent or solidifying agent during the preparation of solid media. Then chemically defined media. So the chemically defined media, it contains nutrient material whose exact chemical composition is known. Whose chemical composition is known is chemically defined media. And this media is used to grow chemoheterotrophs. Chemoheterotrophs means, chemo means the organisms which uses chemical compound as a source of energy are chemotrophs. Heterotrophs, the organisms which cannot use carbon dioxide as a source of carbon and they uses other organic compound as a source of carbon. So those are chemoheterotrophs. And these chemoheterotrophs, they must use the nutrients that containing organic source of carbon and energy in the form of glucose or starch. And this also contains amino acids, vitamins, and other important building blocks required for microbes. So the chemically defined media also contains amino acids and vitamins. and it is not widely used, and this chemically defined media is expensive media. Next, complex media. Complex media is the nutrient media whose exact chemical composition is not known is complex media. This complex media is widely used for heterotrophic bacteria and fungi, and it is made up of extracts from yeast, meat, plants, protein digest, etc. And the composition may vary slightly from batch to batch. And it contains energy source, carbon source, nitrogen source, sulfur source, which are essential for the growth of the bacteria. And it also contains the protein in the form of peptone. Apart from this complex media also contains vitamins and organic growth factors, which are provided by meat and yeast extracts. And the two major groups of complex media are nutrient broth, which is a liquid medium, and nutrient agar, which is a solid medium. So both these contains the same ingredients as nutrients, but a nutrient agar, additionally it contains agar which acts as a solidifying agent. Next, anaerobic growth medium. Anaerobic growth medium is for the cultivation of strict anaerobes or obligate anaerobes. So the anaerobic bacteria are the organisms which grows in absence of oxygen. That is, oxygen is toxic to those bacteria. So the media which is used for the growth of anaerobic bacteria, it contains a reducing agent, that is known as reducing media, and it contains ingredients that chemically combined with oxygen and remove it from the medium. So this reducing media, it contains certain chemical agents which on combination with oxygen, it removes the oxygen from the medium and makes the medium as anaerobic. And the example for anaerobic growth medium is sodium thioglycolate medium. Sodium thioglycolate medium. So here the tubes are heated shortly before used to drive off the oxygen, that is pre-reduced medium. So after preparation of the medium, as the base of the media preparation is water, it may carry some of the oxygen. So when you heat the medium for certain period of time, it will eliminate all the dissolved oxygen present in the medium. Then it has been poured into the plates, that is Petri plates, and in maintaining an anaerobic condition, that is anaerobic chambers has been used for pouring of the medium and as well as inoculation of the organism into the medium. Next, selective medium. The selective media is which is used to suppress the growth of unwanted bacteria, and simultaneously it encourages the growth of desired bacteria, that is selective media. And example for selective media are Sabouraud's dextrose agar media, so which pH has been maintained at 5.6. And this media is exclusively for the growth of fungi. So the low pH 5.6 will not allow the growth of most of the bacteria. So it is not suitable for the growth of bacteria, but it is a specific medium for the growth of fungi. Next, brilliant green agar medium. This brilliant green agar medium, it contains a green dye. This green dye which selectively inhibits the gram-positive bacteria, and this is used to isolate gram-negative bacteria like Salmonella typhi. And similarly, another type of selective media is bismuth sulfite agar medium. This bismuth sulfite agar medium is used to isolate Salmonella typhi. This bismuth sulfite agar medium, it reduces sulfate to sulfite, and thereby it inhibits the growth of most of the bacteria. And this is a specific medium for the isolation of Salmonella typhi organism. Next, differential media. Differential media is used to distinguish the differential characteristics or to bring out the differential characteristics of the bacteria. So it is used to distinguish colonies of the desired organism. And example for this is blood agar medium. So this blood agar media is used to distinguish bacteria that destroy red blood cells, that is hemolysis. The bacteria which will cause hemolysis will produce a clear area at that area where the bacteria has been grown, and that clear area is denoted as a zone of clearance. And the example for hemolytic bacteria is Streptococcus pyogenes. These Streptococcus pyogenes, when it is cultivated in blood agar medium, it causes the lysis of all the RBC cells. So where the RBC cells has been eliminated in the medium, there a clear area has been obtained. So that clear area is known as hemolytic zone. And the next media is both selective and differential media. So this media contains both the properties of selective and differential media. That means which is used to distinguish the colonies of the desired organism and as well as inhibit the growth of the other microbes. And example for this media is mannitol salt agar media. This mannitol salt agar media is used to distinguish and select Staphylococcus aureus. The media pH has been maintained, and high salt concentration 7.5% of sodium chloride has been added in the medium. So that is why it is called as salt agar, mannitol salt agar medium. Addition of the high concentration of the salt 7.5%. This addition of the salt, it discourages the other bacteria which cannot survive in the high salt concentration. And it is also added with a pH indicator which changes its color when the mannitol is fermented to acid. So if the bacteria is able to ferment the mannitol, what happens? The fermentation products are acid and the gas. The acid production is detected by the change of the color of the media. And then McConkey agar media. This McConkey agar media is used to distinguish lactose fermenting bacteria and non-lactose fermenting bacteria. And this is also used to distinguish and select Salmonella organism. And it contains both the bile salts and crystal violet, and which discourages the growth of the gram-positive bacteria. And this media contains lactose along with a pH indicator. So the lactose fermenters will produce pink or red color colonies, whereas the non-lactose fermenters, they will produce colorless colonies. Based on the fermentation of the lactose, the bacteria has been distinguished or differentiated here. The lactose fermenting bacteria, when they are cultivated in McConkey agar medium, they produce pink or red color colonies. So if the bacteria is non-lactose fermenting, when it is cultivated on McConkey agar medium, it produces colorless colonies. Next, enrichment media. This enrichment media is used to favor the growth of microbes that may be found in small numbers. So it enhances the growth of the wanted bacteria. That is enrichment culture medium. So this, unlike selective medium, it doesn't necessarily suppress the growth of the other organism. Selective media enhances the growth of the wanted bacteria by suppressing the growth of unwanted bacteria. But unlike this media, the enrichment media enhances only the wanted bacteria. So it is used mainly for fecal and soil samples, and after incubation in enrichment medium, the greater number of organisms increases the likelihood of positive identification. So this, if the sample contains low concentration of organism or low number or small number of organisms, when they are cultivated in enrichment medium, their growth has been enhanced. That is why it is known as enrichment media. So these are the references for this topic. Thank you for your patient listening.